pleasure and privilege to introduce Professor Jack Rutledge, who's not only one of our senior medical scientists aboard CBST. Yvonne, do you know him?
Yeah.
Oh, okay, fine. So then this is kind of redundant, but Professor Rutledge is also Chief of Division of Endocrinology, Clinical Nutrition, and Vascular Medicine
yes.
at UC Davis. So given his stature and status, he's allowed to talk about anything he pleases, but I think he's going to talk about lipoproteins today. All right, so thank you.
Thanks. So last spring, Yvonne, if you've heard this talk before, you can go. But last spring,
too many talks over. You have to stick around for one.
Last spring, Tom Jew in biological chemistry and a couple of people in biochemistry here put on a conference called Biomembranes Conference. And so they asked me to speak at it and asked a number of us to speak at it. And from that talk, we've written a chapter which will be published in the textbook of biomembranes, something like that. So I thought I'd just go ahead and do the same thing. It gives you an overview of what we're doing in the lab, primarily what we're doing with Thomas Husser and a number of other people. So let me just go back to this. So this is really a combination of the work that the group of us have done over the last number of years. So it's Thomas Husser, who you all know, John Voss, who's in biological chemistry, Atul Parikh, who's in applied physics, and James Chan, who's also at CBEST. So this is the accumulation of the work that we've done probably over the last 5 years. And so this is what I'll talk about over the next 40 minutes or so. Triglycerides and inflammation, single lipoprotein analysis, apolipoprotein E conformational changes in the postprandial state. So postprandial is after you eat. Fasting is before you eat. Preprandial is before you eat. Postprandial is after you eat. And then triglyceride-rich lipoproteins and endothelial cell inflammation, and lipolysis products of triglyceride-rich lipoproteins and monocyte activation. Thanks. So some of you have seen this slide before. One of the things that we do is we wonder about how we go from the food we eat to having heart attacks, to having hepatic steatosis. How do you go from the food you eat to having a chronic disease like atherosclerotic cardiovascular disease or non-alcoholic steatohepatitis? So this is a figure from a study that was done a number of years ago looking at the triglycerides. So triglycerides are one of the lipids that you have in your blood. And looking at triglycerides in healthy college students after they eat a single moderately high-fat meal. And so what you see here is that the triglyceride rises from roughly 150 to roughly 250, so it's about a 60% increase in triglycerides over the 3 hours when you eat a meal. The other lipoprotein that you're probably aware of is low-density lipoprotein. So that's the bad one. Everybody talks about low-density lipoprotein being the bad one. So in the same period, low-density lipoprotein might go up a little bit like this. It may go up 10 points. Sometimes it'll even go down. So after a meal, clearly the most dynamic lipoproteins are the triglycerides. And this is in healthy people. And people who have disease, people who are overweight, who are obese, who have diabetes, it goes up much more dramatically than that. So how do triglycerides cause inflammation? And I would extend it not just vascular inflammation, but hepatic inflammation, perhaps brain inflammation. So sorry about this. I don't know exactly how this happened going from a Mac to a PC, but anyhow, so this is a figure of the same group of people, or a similar group of people, who we gave this moderately high-fat meal to, and then we measured intracellular monocyte tumor necrosis factor. Okay, so monocytes are cells in your blood. They're immune cells. They're very important in diseases like atherosclerosis, like NASH, like non-alcoholic steatohepatitis, fat. They're important in obesity. And so what we found was after giving this single meal to healthy people that the monocyte tumor necrosis factor is a pro-inflammatory cytokine. It's associated with inflammation. It's associated with injury. So monocyte TNF went up about 5-fold or so and never really returned to control after this single meal. This is the model that we have that Colin's been working on, a number of us have been working on. And so just to sort of take you through the biology of this. So this is an endothelial cell. Endothelial cells are the very thin, pancake-like cells that line the blood vessels. They're extremely thin, very flat. They kind of look like a pancake. And they're in control of a lot of the biological functions of blood vessels. They let nutrients in and waste products go back into the blood. Right here is an enzyme that's anchored to endothelial cells, lipoprotein lipase, LPL, and it serves the function to hydrolyze triglyceride-rich lipoproteins. The reason they're called triglyceride-rich lipoproteins is, as the name implies, they're very rich in triglycerides. So this would be like a chylomicron, a very low-density lipoprotein. Low-density lipoprotein has very little triglyceride in it. So this is triglyceride-rich lipoprotein. On the surface of triglyceride-rich lipoproteins, and all lipoproteins for that matter, is the protein species. And I just listed one here. This is apolipoprotein E. So these are proteins that are sort of lying on top of the lipoprotein. So lipoprotein lipase enzymatically hydrolyzes triglyceride-rich lipoproteins to form these subspecies. Remnant particles, the smaller particle of this, fatty acids, phospholipids, monoglycerides, and diglycerides. So this array of lipolysis products can then have effects on monocytes that are in the blood. They can double back and have effects on triglyceride-rich lipoproteins. They can affect endothelial cells, and then they can go beneath the endothelial cell and affect cells, more tissue-specific cells, like the macrophage in blood vessels, the hepatocyte or the stellate cell in the liver. So this is the basic model that we use to explain what we do in our laboratory to people. So this is a triglyceride-rich lipoprotein. As I said, the apolipoprotein is sort of lying over the surface of it. The lipoprotein is on the center. The hydrophobic part of the lipoprotein is cholesterol and triglyceride. On the surface are the proteins, the phospholipid shell, and unesterified cholesterol. So this is one of the first experiments that we did that James and Debbie Moten, myself, Nancy Keim, and Thomas did a while back, and this was imaging individual lipoproteins. So in the past, what we've done in sort of clinical cardiology, clinical vascular medicine is you basically draw the blood, you measure the cholesterol in the mass of lipoproteins in the blood. So you're measuring millions of lipoproteins. You're measuring the cholesterol in millions of lipoproteins. So the difference in this is that we're making our measurements on a single lipoprotein. And the way you do that is you isolate your triglyceride-rich lipoprotein. You put it on a glass cover slip. You image it with a microscope, and with this laser beam, you capture it. You want to get it?
That's okay.
You capture it with the laser, and then this is actually an image of a single triglyceride-rich lipoprotein in solution. And these are the different, this is the Raman spectra. Most of you are more familiar with this, of the triglyceride-rich lipoproteins. And the important parts is this peak right here at about 1680 or so. This peak right here, roughly 1300, which are unsaturated bond peaks. And these saturated bond peaks, which are less than 1200 here. So you look at the ratio of these to see how a particular treatment modifies the saturated to unsaturated bond peaks. That's basically the way you do the analysis.
You might have said this in the beginning, and I've read it many times. Did you say where they're from?
It's from a single lipoprotein.
And it comes from the diffraction light that goes into it?
Right. Yes.
Yeah. So you're shining laser light onto a single lipoprotein particle, and the light, inside will bounce off of all these different vibrational modes, chemical bonds, chemical groups that are vibrating. So causing a change in the energy of that photon to measure that energy surface. So that's basically that. So the spectrum basically is a composite of all of the different interactions that are going on between the light and the particle.
And it's near infrared, right? Yeah. This is the setup. You've probably seen this before, and you'll probably see it again when Thomas talks to you. Okay, so this is actually a clinical study. This was done on people. It wasn't done on molecules or anything. It was people, and these were actually an understudied group. So it's a group of women, overweight women, premenopausal overweight women at mid-cycle, during their menstrual cycle. And so we gave them the same meal. And so this is the composite of the Raman spectra for one subject. So it's measuring hundreds of lipoproteins at each time point. Here's 0, 3 hours, and 8 hours. And I think that some of the things that are most dramatic about it is the saturated bond peaks are much sharper and higher, whereas the unsaturated bond peaks are, I think this one right here, it's a little bit higher too, but not relative to the saturated bond peaks. This was an experiment where we treated a small group of lipoproteins with lipoprotein lipase, the enzyme that induces this hydrolysis. And so this is the fasting VLDL. This is this lipoprotein. Fasting VLDL, we induce lipolysis. And you can see there's a pretty dramatic difference here. This is the background LPL. So that's here and here. But the difference between this and this shows that that particular small group of lipoproteins become much more enriched in saturated fat. Here's some other experiments that we did. We took specific fatty acids, stearic acid and oleic acid, and we compared them. So stearic and oleic. So oleic's a monounsaturated fatty acid. Stearic is a saturated fatty acid. Let me see if I can figure out what the difference is here. I think it's primarily this part right here, showing a difference between a saturated fatty acid and both of these are saturated fatty acids. So that's the reason they look so similar. Stearic acid and palmitic acid are both saturated fatty acids.
You guys all have the chemistry background to know the difference between saturated and unsaturated. So these are all...
You can hardly see it, but oleic acid is up here. This little line right here, going like that. That's oleic acid. So this, and this is different than the two saturated fatty acids. Okay, so we conclude from this that saturated and unsaturated content of individual lipoproteins can be assessed by laser trapping Raman spectroscopy. We're really excited about this. We think this is a really neat thing. We haven't gotten the rest of the world on board yet, but we think it's a really neat thing because it seems so prehistoric to us that people are measuring millions of lipoproteins when they can measure individual lipoproteins in series and have a much more accurate assessment of the lipoprotein content.
So is measuring individual TGRL equivalent to doing a bulk measurement, or you get more information out of looking at individual TGRL?
Well, what you would do with measuring individual TGRL, you'd see if there were subpopulations.
Right. So any heterogeneity,
Right. And that's not all you can do. You potentially can measure oxylipids present. You can measure triglycerides present. So I've just talked about saturated and unsaturated fats. But there are other measurements that you can make that would give a lot more information.
Can you say something about what is the conventional method for doing experiments? How would you make measurements about lipid content? Without using this?
Yeah, you'd measure triglyceride and cholesterol.
With doing what? Like
enzymatic assay.
chromatography? Or
no, it's not. Remember, Colin? It's an enzymatic.
So you use LPL to digest away.
Right, but you don't usually use that in measurement of cholesterol and triglyceride. I think it's an enzymatic assay. Yeah. Oh, yeah. You can measure it in one of these point-of-care autoanalyzers.
So typically, if you're going in for a physical exam and you draw your blood, that would be the technique that they would use to get the right.
Okay, so let's talk about apolipoprotein E. So remember, that's the protein that is over the lipoprotein, and we'll talk about what changes in the fasting and the postprandial state. So apolipoprotein E is associated with atheroprotection, meaning it protects you from developing heart disease. Apolipoprotein E4 is associated with a tenfold greater risk of Alzheimer's disease and a fourfold greater risk of atherosclerosis. The only difference between ApoE3 and ApoE4 is a single amino acid. Just a single amino. That's the only difference, and it changes the conformation. So this is looking at ApoE3 right here. This is the presumed conformation of it, and this is the conformation of ApoE4. So at rest, ApoE3 is a more kind of linear molecule, and ApoE4 is more grouped, the more clustered protein. So in collaboration with John Voss, we used a technique called site-directed, spin-labeled electron paramagnetic resonance. It analyzed the protein structure at the level of the background folds. What you do is you label the protein with these sulfhydryl compounds, which are nitroxide compounds, which label the sulfhydryl-specific paramagnetic nitroxide compound. So what it does is that you label the lipoprotein with two of these nitroxide compounds, and you look at the distance between them. Essentially, that's what you're doing with the electron paramagnetic resonance. You're looking to see how far apart these spin labels are. And this, again, was a clinical study where we took normal people, we gave them apolipoprotein, we gave them this moderately high-fat meal, we engineered this apolipoprotein E4 and apolipoprotein E3, we looked at the effect on on electron paramagnetic resonance, fasting, and in the postprandial state. So if you look at the black, that's fasting. Okay, you can see it kind of right here. It's the most narrow. If you look at ApoE4 at 3 hours, that's the red. It has the broadest sort of distribution. And then at 6 hours, it's sort of intermediate between the two. I won't get into the physics behind this, but basically what this suggests, what it indicates, is that the ApoE4 is going from this more clustered, grouped molecule to a molecule that's more opened out. I mean, it's become a more linear molecule on the basis of eating a single meal. So it's got this dynamism. After eating every single meal, it's in a way it's like a mouth opening and closing. So you have a much more linear apolipoprotein E4. So we think that this potentially can provide some information about why apolipoprotein E4 is a much more pathogenic protein than is ApoE3, which is actually atheroprotective.
Do you have equivalent data? So for ApoE3?
ApoE3 doesn't change. It doesn't change in fasting in the postprandial state. So E3 kind of stays this linear molecule that's sitting on top of TGRL, whereas ApoE4 is more grouped or clustered, and then it opens up in the postprandial state and then closes again 6 hours after the meal.
So my question is, as you said before, that the FO4 that's surrounding this vesicles, does it actually mean that the vesicle basically increased, and by these...
It's not a size change. It's a conformation change. You would think, well, if the protein gets bigger, you might stretch the ApoE4 out over the top of it, over the basketball.
Yeah.
But what happens is that at this point in time, the lipoproteins, the TGRL, are starting to get smaller at 3 hours. And we've done with vesicles, and it doesn't appear to be primarily related to the size of the particle. Again, sort of illustrating from a paper we wrote about this. Here are the two nitroxide labels in apolipoprotein E4, fasting, and then postprandial. Separates it, becomes more, there's a loss of this interaction here, actually. I think I'm going to skip over this. Okay. So what we conclude from these experiments are that VLDL lipolysis products increase fluidity, that's what I skipped over, of the particle, and ApoE4, and it induced ApoE4 conformational changes. So we think that the reason this happened was that the fluidity of the surface of the triglyceride-rich lipoprotein increased, allowing the ApoE4 to sort of move apart, these nitroxide labels to move apart.
Okay. Can lipoprotein particles form spontaneously in a test tube if you mix all the components together?
Yeah, you'd form micelles.
And so would there be, if you had a vial containing these things, would there be exchange of triglycerides between them?
Yeah. Yeah. And within the body there is a lot of exchange of lipids. So there's a class of enzymes called cholesterol ester transfer proteins, and there's another class called triglyceride transfer proteins. And so there's this shuttling of cholesterol and triglyceride between lipoprotein particles that's happening all the time.
So do you do anything to maintain stability of your
to prevent that?
Again?
Well,
say, for example, you draw blood out, but then you don't look at it until 5 hours later. Would there be a difference between looking at that and looking at somebody who you drew that blood from that patient 5 hours later?
Yeah, well, we try to do it as soon as we can, but there is some delay. I mean, just because you have to run the samples pretty much together. So there is some delay, but what we, in terms of particle size, fits everything that's been published in the literature.
So there are no enzymes that are involved in building these systems?
Not as far as we know. We can't really detect that. If the particle size is not changed, if the cholesterol content is not changed, comparing the two techniques, I don't think that's an issue. All right. So now I'm going to talk a little bit about how these lipolysis products may affect the endothelial cell. So this was a study we did a number of years ago where we perfused the blood vessel of a rat. So the artery here in a human or a rat is called the carotid artery. So we took the carotid artery from a rat and we perfused it with a fluorescently labeled triglyceride-rich lipoprotein. So what you see up here at the top, this is the blood vessel. If a blood vessel is like a tube, we took a section like that, and you're just seeing the very bottom part of it. So this is called a coronal section of the blood vessel. Right here is the endothelium. Beneath that is the smooth muscle cells, and then you've got this adventitial tissue on the outside. So we perfused with just the VLDL, or just the triglyceride-rich lipoproteins. We add lipoprotein lipase. You can't really see too much with just white light. However, if you look under fluorescent light, what you see is that when you perfuse the triglyceride-rich lipoprotein alone, there's very little accumulation in the blood vessel. If you perfuse with lipoprotein lipase, you see this intense staining right on the surface of the blood vessel. And this is the overlap of the two right here. So it's right on the endothelial surface. This is one of the earliest manifestations of atherosclerosis, is an increased uptake of lipoproteins on the surface of the blood vessel. Okay, lipid rafts. Are you guys familiar with lipid rafts? Okay. Yvonne, are you familiar with lipid rafts? Okay, so you've probably heard Dr. Parik's talk. Or maybe not. So we did some experiments with Dr. Parik where we took endothelial cells and we labeled the lipid raft with a cholera toxin. And it's relatively specific for the lipid raft and plasma membrane. We also labeled the VLDL with an ATO dye. It's a molecular probes dye. It lights up red. And then we did a merge of the two. So if you treat an endothelial cell with VLDL alone, there's this kind of co-localization here and maybe here. However, if you treat with VLDL and LPL, thanks, Frank. What you see is this, and then this is the overlap of the two. So in our paper, we made the case that when you induce lipolysis, that lipid rafts tend to aggregate. Okay, they tend to become bigger and aggregate, and that VLDL accumulates in the same area as the lipid rafts. We were thinking presumably on the lipid rafts. This was interesting to us because it showed two things. One, that lipid rafts tend to aggregate with VLDL lipolysis products, and that the VLDL remnant particles tend to accumulate on the lipid rafts. Carrying this out a little bit further, Larissa Esseline in our lab did some experiments where what she did was she wanted to measure permeability with VLDL lipolysis products, which she did using a couple of techniques. These were some experiments where she stained endothelial cells. So endothelial cells are the main permeability modulating cells in blood vessels. And so what Larissa did was she looked at a number of these junctional proteins. So that one of them is ZO1, another one is occludin, another is VE cadherin. You can see the endothelial cells here. Here's the nucleus, here's the cytoplasm, here's the junction between the two endothelial cells. So this is treatment with media. This is treatment with TGRL. You can see there's this nice staining of the junction between the endothelial cells. This is LPL alone. And then this is TGRL plus LPL, the induction of the lipolysis products. And what's different about this is that the junctional proteins, say here, are rearranged. They're more perpendicular to the cell, if you will. And you see that also over here to some extent with VE cadherin. You can't really make too much out of it with the occludin. At the same time that we were able to document that there was a change in these junctional proteins between endothelial cells, that was also the time of the peak increase in permeability. So we think that the permeability increase is related to the cell junction between adjacent endothelial cells. Okay, we'll talk about monocytes now. We've done a number of experiments now in monocytes and also in hepatocytes, and we see something that's actually really quite consistent. Another cartoon that I think Samantha Ford developed a while back, where you're familiar with this down here, you're familiar with these lipolysis products. The lipolysis products have effects on circulating monocytes, and to our surprise, and as a result of Samantha's sharp observations, what she saw was that if you treat monocytes with these lipolysis products, they form lipid droplets. okay, in blood after a meal. So you eat a meal, you really don't have many lipid droplets in the monocytes. After the meal, lipid droplets form and are present in the blood. What we don't know yet is what the functional outcome of these lipid droplets are. Does that mean they're activated? Does this mean they're more likely to bind to the endothelium? We don't know right now. So this is the approach that we took. Again, using the laser trapping Raman spectroscopy. You trap a monocyte, and you can develop a Raman spectra of the monocyte. And this is maybe the first experiment that we did with this. So we treated the monocyte with media, with LPS, and with VLDL plus LPL. These are the Raman spectra. So with media and LPS, you see that there is a change in the topography of the monocyte. It has a change in the topography. And if you look at the Raman spectra, you can see maybe there's a little bit of difference. Just looking at it, big picture, there may be a little bit of difference. However, if you treat those monocytes with VLDL and LPL, you see these lipid droplets that form throughout the monocyte. And you see over here there's a fairly dramatic change when you compare it with, say, the LPS treated or the media treated. I'm sorry, I didn't define LPS. LPS is lipopolysaccharide. It's a component of the cell membrane, a bacterial cell membrane. It's associated with sepsis. Okay. So far and away, the one that caused the most perturbation of the monocyte was the VLDL plus the LPL.
So is this what you call activated? Or do you consider this to be activation?
I would, but we really haven't had the time to do the experiments to see if it sticks more. And this is using CARS, coherent anti-Stokes Raman scattering. Yeah. So I'm not even going to attempt to do this. I'll let Thomas and Frank tell you about this, but these are the lipid droplets that you can see. And by focusing on a single lipid droplet, you're able to characterize that lipid droplet over time. So the way we would measure the content of this lipid droplet is we would kill all the cells, float up the lipid, and measure the lipid that you extract from the cells. So using this technique, you're able to image the cell, and you're also able to assess the contents of individual lipid droplets over time without killing the cell. Here are just some more pictures of it, looking at some of these saturated and unsaturated bond. These are some of the lipid droplets that we see, and it's very predictable. It's predictable with hepatocytes. It's predictable with stem cells. Pretty much every sort of mesenchymal derived cell has the same effect. This is actually a monocyte that's untreated, and you can see relatively few of these lipid droplets. Here are a few more. This is one with these lipid droplets here, and then I think this is CARS right here. Just give you a few more examples. And it's amazing to me that this can happen that fast. So if you have the cells in front of you, you treat them with VLDL and LPL, 30 minutes, maybe 50% of them have lipid droplets. And then at about 3 or 4 hours, about 10% of them have died. But it looks like a lot of the cells have been able to adapt to this and have somehow or another gotten rid of the lipid droplets. So it's not apoptosis per se. There's more to it. It's not that the cells are dying and they're developing these blebs, these apoptotic blebs. It's that these are lipid droplets that have the ability to grow and expand and retract. I was talking with a relatively well-known GI gastroenterologist, And she was saying that in their work, they see that these lipid droplets form quickly in the liver. I mean, it may be exactly like what we're seeing here, that after a meal, in your liver, you form these lipid droplets that are metabolized in the hepatocyte, and then before another meal, you have relatively few lipid droplets.
How large are these droplets compared to with respect to a single lipoprotein bubble?
Oh, it's not a single one. And we don't even think it's lipoproteins. We think it's probably fatty acids. These things form so quickly, it's just impossible for lipoproteins, which are so big, to get in the cell that fast. So we think it's through we think the lipid droplets are composed of fatty acids. The question is, how do they get into the cell? We're not sure about that yet.
Do you think they're already there?
Well, people have postulated that before. I guess it's possible, but this is an enormous amount of new lipid in the cell. I mean, if you just look at the volume of this, maybe 20% of the volume of that cell is new lipid. So it's kind of hard to see how it could be released from structures within the cell.
But most things probably indicate that it's actually should come outside from the cell.
From the outside in.
Yeah, because it's usually just happens if you treat it with VLDL and LDL together.
Yeah.
If you treat it singly with one order matter, it doesn't change anything. But it changes a little bit. And you can see
if you treat with VLDL, you don't see any of this.
Not really droplets, but I actually just analyzed some droplets in hepatocyte cells, also with the same treatment, and you could see some little changes if you just treat it with VLDL. But
you're right.
Dramatic. And you don't see, or you barely see the creation of new droplets.
Yeah.
But for this one,
I think it's got to be from the outside in. I don't see how it can be released from, mitochondria or something in the cell. I don't see how it can be that.
Yeah, and actually some studies about free fatty acid influx inside the cells, and also the creation of the droplets.
So we did some experiments trying to look at endocytosis as a mechanism. And we used different sizes of dextran to see fluorescently labeled dextran. And there was just no correlation. I mean, we didn't really think it was endocytosis. Now, it could be this sort of flip-flop mechanism that people talk about. That could happen relatively quickly. And correct me if I'm wrong, but I think the way this works is that you have fatty acids are in the plasma membrane, and the fatty acids can come and integrate in the membrane, and then flip to the other side, and then flip out into the inside of the cell. Is that how you understand?
Right. But actually, even discussions about that, because they made some studies, because that would be basically kind of like a diffusion process, and they measure that out, and they get like two constants out, like one which would indicate that this is diffusion, but they actually overlaid with another constant which kind of indicate that there may be some proteins or like transporter proteins which bring them also.
That are chaperones.
Yeah, so they're basically like two different ways how they actually enter the cells.
Well, I hope the person who reads our grant doesn't know about this other theory. Just a few more pictures of this. Okay, so we concluded from this that individual monocytes can be analyzed by laser trapping Raman spectroscopy, and lipolysis products injure the monocytes. Laura Higgins, who finished her PhD in my lab, has 2 whole papers looking at the effect of lipolysis products on monocyte function, and it's very interesting. And she also includes some of the ApoE work, which was surprising to us. So, what I've covered over the last several minutes is that the lipid saturation of individual triglyceride-rich lipoproteins can be assessed by laser trapping Raman spectroscopy. Lipolysis products can have effects on the apolipoprotein E3 and E4. We have further data to indicate that apolipoprotein E3 can bind a number of potentially toxic products in the blood, so it may act as sort of a scavenger for other toxic products in the blood. VLDL lipolysis products appear to injure the endothelial cells. We think that this is probably through a cell signaling mechanism that may involve some relatively esoteric processes, SMAD signaling, and a transcription factor, activating transcription factor 3. It's too hard to go into that. It's pretty complicated, but we have pretty good data there. The VLDL remnant particles co-localized with remodeled endothelial cell lipid rafts, and individual monocytes can be analyzed by laser trapping Raman spectroscopy, and you can image the individual lipid droplets with laser trapping Raman spectroscopy and CARS. So if you've been in Davis for a relatively short period of time, you haven't seen this except spelled the other way. Redrum Burger over by In-N-Out Burger. This is the way it was. This was originally the way it was. It was Murder Burger. Okay.
So, has anyone tried to develop like C14 labeled lipids or things like that?
We're doing that right now. We're doing that. Actually, we're doing it with glucose.
So you mean glucose? You start with C14 glucose, and then that gets used to.
No, I'm sorry, not C14. Deuterated glucose. Deuterated glucose. We haven't done other labs have done that, and it's a very cumbersome, difficult process. So you label the lipoprotein, you inject it into an animal, in some cases into humans, and you look at the metabolism. But it's a very difficult thing to do, and it's basically an exercise in mathematics, which has always scared me.
But even with so anything with a different mass should result in a unique Raman peak. So still using the same techniques, then you'd be able to distinguish between exogenous lipids versus stuff that is endogenous.
Right.
Have you actually also conducted some treatments with pure free fatty acids?
Pure?
Like just free fatty acids, not in triglyceride form, basically. Have you done something like that?
Have not. We should work on it together.
So you're basically taking the triglyceride, taking
just
fatty acid tail.
Exactly.
That would go after cholesterol.
Yeah.
No, I haven't done that. So you've been working with Thomas on. Have you been working in hepatocytes now? Now we've got some. There's some really interesting work going on in that. Yeah. So are you writing up your paper?
Yeah, I've kind of like collected data. The problem is, we can really, with the hepatocyte, the observation of the lipid accumulation is much bigger, as you probably saw in the videos. But the problem is, we struggle a little bit with the change in focal change. So if we would just publish it as it is right now, like everybody would just like sort of say, you have a change in the focal, maybe you just focus in another area, and that's why you can see this lipid up there. So we need a little bit more around it.
To explain to the rest, so we developed a product after the results of these monocyte experiments. We thought, what the heck? Let's just throw some VLDL and LPL on some hepatocytes and see what happens. And as Ivan said, it's a much more robust pattern in hepatocytes. And so we're hoping we can write this up pretty quick, because it explains a lot in terms of this condition. There's two conditions. One is called NAFL, non-alcoholic fatty liver disease. So if you eat too much fat, if you're overweight, if you have diabetes, you develop a fatty liver. So the liver becomes interspersed with fat. It's like foie gras. You feed a duck fat and they develop a fatty liver. Same thing happens with humans. And then the extension of that is that after a period of time, some of the people who have this fatty liver go ahead and develop inflammation of it. That's called NASH, non-alcoholic steatohepatitis. And then a certain proportion of those people will go ahead and develop liver failure and cirrhosis. So it's becoming the most common cause of cirrhosis in the country, exceeding alcoholism. So it's becoming a really important problem. And we think we've got a nice handle on what's going on here. That it's these fatty acids that are inducing, these lipid droplets, and it's very dynamic. It goes and comes. I mean, one of the interesting things to do is to see if we can figure out what happens to the droplets once they're in there. What happens? Because the cells don't die. I mean, a proportion, about 10% of them die, but not all of them die. And so there's got to be some humongous, scavenger mechanism that's taking the lipid out, distributing it better in the cell.
That's what I also thought. Just to treat the cells a little bit, the accumulation of lipid droplets is not enormous, but visible. And take all the media out and just leave the cells as they are with the lipid droplets. And it would be interesting to see if they would actually use the energy which is stored in the lipid droplets actually to kind of like sustain further. So that would be...
And we had quite a bit of other confirmatory data too. So if you looked at measures of hepatocyte inflammation, like reactive oxygen species formation, it went up quite a bit. Looking at inflammatory genes, they went up quite a bit.
Do most fatty liver cases progress to...
No. In fact, if you're obese, there's a very good chance you have fatty liver, at least at some time. But it's only a fraction of those. So a fraction of obese people will have fatty liver, a fraction of people with fatty liver will have NASH, a fraction will develop cirrhosis. But it is becoming the most common cause of cirrhosis. And it always used to be alcohol was the most common cause of cirrhosis. Now it appears to be NASH.
Is the rate of optimal cirrhosis
Say again?
Is the rate of cirrhosis due to alcohol constant?
Yeah, it's not gone down any. There are as many alcoholics now as there were 20 years ago, as far as I know.
The thing is, you want to develop that due to whiskey or alcohol. If you start to drink or to eat enormously, it's going to disappear. It's going to recover again. So the lipid droplets, basically, the accumulation of the droplets, it will go after a while. It will recover.
You recover if you start drinking again?
No. If you stop drinking and if you stop eating a lot, then your liver starts to recover. So it's not a permanent state. So it's not, if it once is there, it don't stay forever. It's not like this.
Once you develop fibrosis, then it doesn't.
Then that was pretty late.
But you see this all the. I'm becoming more and more aware of how common this is. Like I said, there are some people who develop these lipid droplets apparently after they eat a meal and then they go away. I was at the heart meetings in November, and they presented. One of the people there presented some data that neonates, that the fetus and neonates of obese women have fatty liver, which is scary when you think that the liver is exposed at that early in life to reactive oxygen species, to all these inflammatory genes. It's really scary to think about.
So to move forward in the research, do you feel that imaging at the single cell level is what you need, or you need to kind of open it up and start looking at things at a tissue level?
Both. We need Ewan to write the paper. That would be the first step. And then after that, but we still need to we need to study it on. We need to study it on the subcellular basis too. It's such a cool thing that if we can characterize individual lipid droplets, there may be a difference in the lipid droplets of people who are destined to develop cirrhosis versus those that aren't. Because it's not all of them. It's only a minority of those people. So there may be something different about the lipid droplets that could tell us a lot about the whole disease process. All right. Got to run back over to the med center.
All right. Yeah,
Okay.
any questions? Thanks, Dr.
Yeah, thanks.